So what if the absolute most sophisticated AI we have for predicting breast cancer treatments is basically useless in a real clinic? Like entirely, because it just can't explain its own math.
Yeah, I mean, it's the ultimate medical paradox, right? We've built these incredibly powerful machine learning models that can, look at a patient's tumor and instantly recommend this complex combination of drugs.
Right.
But to an oncologist, a recommendation without an explanation is, well, it's just a guess. If you can't actually see the biological mechanism driving that prediction, you're not going to trust it with a human life.
No, of course not. And when you step into the reality of treating something like breast cancer, trust and understanding are everything because the battlefield it's constantly shifting.
Oh, absolutely. It's a highly dynamic environment.
Right. We're not dealing with a static target. The moment therapeutic pressure is applied, like the moment those drugs enter the system, the cancer cells begin rapidly rewiring their own internal supply lines.
They do.
They change their signaling networks just to survive. And that's the primary driver of drug resistance, isn't it?
Exactly,
Yeah.
which is the fundamental reason modern oncology relies so heavily on combination therapies. Like hitting the tumor from just a single angle, it just prompts it to reroute.
It just finds a detour.
Right. But if you hit it with multiple drugs simultaneously, aiming at different vulnerabilities, you can theoretically trap the cell before it has a chance to adapt. The real challenge is the math.
Because there are so many combinations.
The number of possible drug permutations is just astronomical, which is exactly why we need AI to sift through them all.
But then, we hit that black box problem. The algorithm spits out, hey, combine drug A with drug B, but it locks the underlying Y inside a dark room.
And no one can see inside.
Exactly. So today, in this deep dive, we are cracking open that room. We're breaking down this really groundbreaking new study that introduces a hybrid AI framework designed specifically to be readable.
It's a totally different approach.
Yeah. It merges something called Boolean modeling with a random forest algorithm. And the mission for us today isn't just to talk about predicting drug synergy, it's to deeply interpret how those drugs are, physically manipulating the intricate signaling networks of breast cancer.
Right. We are essentially transitioning from AI as this blind oracle to AI as an active partner in biological discovery.
Which is huge. So I want to start with the first half of that hybrid framework, because simulating the biology of a cancer cell sounds, frankly, impossibly complex. How are they taking something as chaotic as a human tumor and turning it into a mathematical model?
Well, they do it by stripping all that complexity down to its absolute most basic binary state using Boolean logic.
Okay. Binary meaning just zeros and ones.
Exactly. The researchers mapped out 117 specific, really critical proteins involved in breast cancer signaling. And instead of trying to calculate the exact, analog concentration of every single protein,
which would require an insane amount of computing power.
Oh, computing power we simply do not have. So instead, they treat each protein as a light switch. A 0 means the protein is inactive. A 1 means the protein is on, or active.
So it's like a binary cascade. If protein A gets turned on, maybe it flips protein B on, which then, flips protein C off. It's simulating the whole chain reaction.
That's precisely the concept, yeah. They took 5 different breast cancer cell lines, including widely studied ones, like MCF7 and MDA MB468.
Right.
And they simulated their specific genetic profiles.
Right.
Then they introduced the digital equivalent of therapeutic pressure.
The digital drugs.
Right. They threw 574 different drug pairs at these simulated cells, calculating 16 different combination doses for every single pair.
Wow.
And they let the digital simulation run until the cascading ones and zeros finally settled into a permanent, unchanging pattern.
Okay, wait, so they just stopped flipping?
Exactly. In systems biology, this is known as a steady state.
Okay, got it. So the Boolean model acts as this biology simulator. It outputs a final snapshot, like a massive map of which of those 117 proteins are ultimately left turned on or off after the drugs have done their damage.
You nailed it. And that snapshot, that steady state, is what gets handed over to the second tool, the random forest algorithm.
Which is basically the data cruncher in this scenario.
Right. The random forest is the pattern recognizer. It takes all those steady state protein activities across thousands of simulations and uses them to predict a highly specific metric called the HSA score.
The highest single agent score. And if I'm remembering the formulation correctly, this is kind of the gold standard for measuring true drug synergy, right?
It is, yeah.
It essentially calculates how much better the combination of 2 drugs performs compared to just using the single best drug of the pair all by itself. Like, does 1 + 1 = 3?
That's a great way to put it. The math is really crucial here. If the HSA score is positive, you have true synergy. The drugs are actually cooperating to suppress the cancer more effectively than they ever could alone.
Okay, and if it's negative?
If the HSA score dips into the negative, you have antagonism, meaning the drugs are actually interfering with each other. They're helping the cancer survive.
Yikes. Okay, but I need to play devil's advocate here for a second, because looking at the underlying data for this random forest model, the pure predictive performance doesn't exactly look like a slam dunk.
It's not perfect.
Right, like the Pearson's correlation is sitting at 0.40, and the area under the curve, the AUC, is 0.67. In the data science world, those are solidly moderate numbers.
They are.
I mean, it has a sensitivity of 0.81, so it's fairly good at spotting synergy, but its specificity for spotting antagonism is way down at 0.39. It feels, I don't know, risky to champion a model that is functionally blind to drugs canceling each other out.
It's a completely valid critique of the raw metrics, but I'd argue it entirely misses the paradigm shift this model actually represents.
Okay, how so?
You have to ask yourself what is actually useful to a clinician. A neural network with a perfect 0.99 AUC is mathematically beautiful, sure, but if it recommends a lethal combination to a patient and you can't audit its reasoning,
You just can't use it.
exactly. The researchers accepted moderate predictive performance as a deliberate trade-off for total interpretability. Because the inputs to this random forest are just the plain readable states of those 117 proteins, we can actually trace the exact biochemical logic the algorithm used to arrive at its score.
Okay, so let me do a quick mini recap here to make sure we're all tracking. We're trying to outmaneuver cancer's ability to rewire its supply lines.
Right.
Bullet point 1. To do that, the researchers built a hybrid engine using Boolean zeros and ones to simulate how 117 proteins react to a drug combo until they hit a steady state.
Correct.
Bullet point 2. A random forest algorithm then analyzes that steady state to predict the HSA synergy score. And bullet point 3. The true value here isn't flawless accuracy, it's total transparency. Which honestly brings up a question for you, the listener. Think about your own field, whatever you do for a living. When is understanding how an algorithm made a decision actually more important than the decision itself?
It's a daily tension in modern medicine for sure, but once you have that transparent map, the real excitement is seeing what it reveals about the biology of the disease on a massive scale. Which brings us to global interpretability.
Yes, let's zoom out to that global view. Because the researchers wanted to know which of those 117 proteins actually mattered to the algorithm. And they used this statistical method called accumulated local effects, or ALE.
Right, ALE.
As I understand it, ALE isolates the marginal effect of a single feature. It basically asks the math, hey, if we hold everything else constant, how much does changing this one specific protein alter the final synergy prediction?
It's a phenomenal tool for cutting through the noise. And out of the 117 proteins simulated, the ALE analysis revealed that just 30 of them accounted for 98% of the model's entire decision-making power.
Wait, really? Just 30 proteins do almost all the heavy lifting?
Just 30. And when you look closely at the biological function of those 30 heavy hitters, they cleanly divide into two opposing factions.
Right, the first faction being the oncoproteins. We're talking about the big drivers like MTORC1, NF-kappa B, S6K1, and PGR. These are essentially the cellular engines pushing for unchecked growth.
The gas pedals.
The gas pedals, exactly.
And the ALE curves for these oncoproteins showed this definitive universal trend. As the activity level of an oncoprotein goes up, the predicted drug synergy, that HSA score, drastically drops.
So more gas equals less synergy.
Absolutely. For instance, the data isolated the PGR protein was just staggering. When PGR activity was low, the model predicted a healthy positive synergy score of 1.31. But as PGR activity spiked, it dragged the entire synergy score down to a negative 2.51.
Oh, wow. So the high oncoprotein activity just completely fractured the efficacy of the drugs.
That ruined it entirely.
Which logically tracks, right? I mean, if the cancer cell is flooding its own engine with survival signals, it becomes exponentially harder for two drugs to coordinate a knockout blow.
Right.
And then you have the second faction, which acts as the exact inverse.
Yeah.
The tumor suppressor proteins.
The brakes. And the most prominent tumor suppressor in that top 30 list was FOXO3. The algorithm recognized that as the activity of tumor suppressors increases, the drug synergy score rises right alongside it.
It's a really straightforward mechanical relationship. Pumping the oncogenic gas lowers your synergy while leaning on the tumor-suppressing brakes raises it.
Exactly.
So here's our second quick recap for you listening. Bullet 1. Global interpretability looks at the overall trends across thousands of simulations. Bullet 2, just 30 proteins do 98% of the work predicting synergy. And bullet 3, our memory tool, gas goes down, brakes bring it up. High oncoproteins ruin drug synergy, high tumor suppressors enhance it.
That's a perfect way to remember it.
So, question for you, the listener. If you were designing a new breast cancer drug, would you focus your billions of dollars on cutting the cancer's gas line or trying to fix its brakes?
That strategic debate is exactly what this global data informs. However, treating a population isn't the same as treating an individual patient.
Right, because everybody's different.
Broad trends are incredibly useful, but human tumors are defined by their unique, highly erratic mutations. So to bridge that gap, we have to move from global trends to local interpretability.
This is where things get really intense. This is where we zoom all the way in on a single biological event, and the researchers deploy this algorithm called tree interpreter.
It's an amazing tool.
Yeah, it literally cracks open a single prediction for one specific drug pair on one specific cell line and calculates the exact numerical contribution of every single protein. So let's walk through this first case study they highlighted, because honestly, it reads like a biochemical thriller.
It really does. So the setting is the MCF7 breast cancer cell line. And the weapons of choice are two drugs, AZD5363, which is designed to inhibit the AKT protein, and fulvestrant, which targets the ESR1 protein.
And under normal baseline conditions in this cell line, these two drugs exhibit massive synergy, right?
They do. They dismantle the cell's survival networks highly efficiently.
But researchers introduced a complication into the simulation. They engineered a gain-of-function mutation into the AKT protein. They basically broke the switch so that AKT is hyperactive, just permanently jammed into the on position.
And the resulting data from that is brutal. The synergy between those two drugs entirely collapses. The HSA score craters by -3.74.
The combination goes from being highly lethal to almost useless.
Completely useless. But thanks to the local interpretability of the model, we don't just log the failure. We can actually watch the biological dominoes fall in real time to see why it failed.
Okay, I want to trace those exact dominoes. What did the model see happening inside the cell once that mutated AKT took over?
The very first thing the algorithm flagged was the suppression of our primary brake pedal, FOXO3.
Ah, the brakes.
Right. In the normal cell, FOXO3 was actively contributing a score of 0.75 to the drug synergy. But the hyperactive AKT directly neutralized FOXO3, driving its contribution straight down to zero.
So the brakes are instantly cut.
The brakes are cut, and then the accelerator gets slammed. The tree interpreter showed that the overactive AKT simultaneously cranked up the activity of mTORC1 and NF-kappa B.
Our gas pedals.
Exactly. This is the cancer cell realizing it's under attack and frantically shifting into a massive structural survival mode. It starts promoting aggressive protein synthesis through a highly specific network of pathways.
Wait, let me guess the pathways.
Go for it.
The 4EBP1, EIF4E, and S6K1 pathways.
You got it.
Let's clarify what that actually means, because those aren't just alphabet soup acronyms. When a cell activates 4EBP1 and EIF4E, it is literally forcing the creation of new ribosomes. It is scrambling to manufacture the physical, structural building blocks it needs to repair the damage the drugs are causing.
It's rebuilding the walls while they're being knocked down. And it creates alternative avenues for growth, too. The model caught AKT upregulating the HES1 pathway, which allows the cell to bypass the drug blockades entirely and just continue proliferating.
Okay, analogy time. Think of the cancer cell as a major city. The two drugs we administered are barricades set up on the main highway to stop the flow of supplies. Normally, that starves the city out.
A blockade.
Right. But introducing this AKT gain-of-function mutation is like giving the cancer a hyper-advanced mutated GPS system. The exact second you block the highway, the GPS instantly routes all the supply trucks through a labyrinth of side streets, routing them through mTORC1, through NF-kappa B, through HES1. The barricades are still there, but the traffic completely bypasses them.
It's a perfect illustration of how dynamic the rewiring process is. The mutation didn't just break the drugs, it built an entirely new infrastructure to render them irrelevant.
So stepping back for our third mini recap. Bullet 1. Local interpretability zooms in on specific drug and cell scenarios. Bullet 2. An AKT gain-of-function mutation totally ruins the synergy of our two drugs here. And bullet 3. It does this by turning off FOXO3, cutting the brakes, and routing traffic through alternative survival pathways like mTORC1 and NF-kappa B.
The side streets.
The side streets. So, question for the listener. Can you think of a time when fixing one problem just forced an adversary to find a vastly more complicated workaround? It's terrifying. You solve the obvious problem and the biology just outsmarts you.
It is the ultimate game of whack-a-mole. But, it's vital to recognize that mutations in cancer aren't exclusively defensive. A mutation can just as easily break a critical structural pillar and open up a massive vulnerability.
Ooh, which pivots us perfectly to the second case study. We just explored how a mutation destroys synergy. Now we look at how a different mutation can actually weaponize a drug combination.
Right.
The setting here is a triple-negative breast cancer cell line called MDA-MB468, and the drugs being tested are a MEK inhibitor and a STAT3 inhibitor.
Now, in the unmutated baseline simulation, this specific combination is pretty underwhelming. The HSA synergy score sits at a meager 1.43.
Which is barely synergistic.
Exactly. And the AI shows us exactly why. The natural underlying activity of mTORC1 and NF-kappa B in this particular cell line provides just enough background survival signaling to blunt the impact of the drugs.
Okay, but then the simulation introduces a gain-of-function mutation into a completely different protein called PTE. And when PTE is hyperactivated, the outcome completely inverts.
Just dramatic.
The synergy score skyrockets, increasing by positive 4.67. The drug pair suddenly becomes incredibly lethal to the tumor. So how does a single mutation turn a mediocre treatment into a devastating one?
Well, the AI traces this beautiful sequential cascade of suppression. Hyperactive PTN acts as a natural overwhelming inhibitor on AKT.
Wait, the same AKT from the last study?
The very same. By crushing the AKT activity, PTN subsequently starves and downregulates EGFR.
And EGFR is the upstream activator for both MEK and STAT3, which are the exact targets of our two drugs. So the mutation is essentially softening up the very targets the drugs are meant to hit.
It softens the primary targets, and then it systematically dismantles the backup systems. By keeping AKT suppressed, PTN also crashes the levels of NF-kappa B and HES1, which breaks that side street escape road we talked about earlier.
No more detours.
No more detours. And crucially, it forces the inactivation of both mTORC1 and mTORC2.
Okay, we've established that knocking out mTORC1 stops the cell from synthesizing new survival proteins. But what is the specific mechanical impact of knocking out mTORC2?
This is where it gets really elegant. When mTORC2 goes offline, the cell loses its ability to suppress NEDD4L.
Yeah, NEDD4L.
Yeah, NEDD4L is a powerful tumor suppressor that relies on ubiquitination to mark oncogenic proteins for destruction. By inactivating mTORC2, the PTN mutation basically unshackles this tumor suppressor, allowing it to sweep through the cell and just accelerate the breakdown of all those survival signals.
Wow. Okay, this feels like it has staggering clinical implications. If an oncologist knows that a hyperactive PTN network acts as a master switch that transforms these specific MEK and STAT3 inhibitors into a highly lethal combination, could they theoretically administer a priming therapy designed to artificially hyperactivate PTN right before they deliver the inhibitors?
You've just described the holy grail of this research. If you can accurately map the network, you can manipulate the network before you strike. You artificially induce the vulnerability, and then you exploit it.
You set the trap, and then you spring it. Okay, final recap for this section. Bullet 1, mutations aren't always bad for treatment. Sometimes they create vulnerabilities.
Right.
Bullet 2, a PTE mutation vastly increases the synergy of ME and STAT3 inhibitors. And bullet 3, it achieves this by suppressing AKT, which sequentially crushes EGFR, NF-kappa B, and the mTOR pathways.
The whole house of cards falls down.
Exactly. So, listener, ask yourself, how might knowing a cancer's specific genetic Achilles heel completely change the order in which we give chemotherapy drugs?
It completely reframes how we might approach treatment timelines.
Yeah.
And that broader perspective is really the triumph of this entire methodology. The field of computational biology is overloaded with black box neural networks that simply output statistical guesses. Give the patient this drug, but this research proves that we don't have to settle for that.
We don't have to stay in the dark.
No. By grounding our AI in the dynamic Boolean simulation of actual signaling networks and parsing those networks with interpretable algorithms like random forests, we are literally decoding the cancer's battle plan.
It bridges the gap between raw data and biological reality.
It does. It aligns conceptually with other deep learning interpretative models like transenergy, but it really stands apart because it relies on dynamic simulations that incorporate a tumor's specific real-time genetic profile. It doesn't just look at a static picture, it watches the cell react.
It's a remarkable shift. But as we wrap up this deep dive, I want to pose a final kind of provocative thought regarding the limitations of this tech.
Oh, there are always limitations.
Right. Because we noted earlier that this specific model is excellent at mapping synergy, but it has a significant blind spot when it comes to predicting antagonism situations where drugs actively fight against each other.
Yeah, the specificity was quite low there.
So, to you listening, if clinical trials begin relying heavily on AI models with this specific bias, how might that blind spot bias the types of therapies that actually make it to market in the future? And based on all the complex pathways we discussed today, the mutated GPS systems, the cellular side streets, the cut breaks, what specific biological detour would you want to ensure is blocked if you were designing your own hypothetical cancer cure?
The terrain is incredibly complex and, no algorithm is flawless. But for the first time, we actually have a map that lets us see the roads.
And that changes everything. We started this deep dive talking about how the microscopic battlefield of cancer is never static. The moment you introduce a drug, the cancer starts rewiring its supply lines to survive. But thanks to interpretable AI, those adaptations aren't happening in the dark anymore. We can trace the detours, we can anticipate the rewiring, and ultimately, we can cut the cancer off at the pass.
Until next time.